Hello everyone, we are the students of Ashok Ramani Institute of Pharmaceutical Science and Research Sapi. Today we are conducting seminar on quality assurance presented by Rupali Ravla, Vaishnavi Nije, Monika Pawar, Aniket Saptale, Prajakta Shilke and guided by Mr. A.N. Dhage sir. So, our questions are first, explain different parts of process validation. Second, explain hydrolytic resistance test for glass container. Third, enlist steps for ISO registration. Fourth, define recall and classify it. What is SOP and quality audits? So, let's begin with the answer. First, explain different types of process validation. Process validation is a critical aspect of pharmaceutical manufacturing and is defined as the documented evidence that a process operates within established parameters and consistently produce a product that meets its predetermined specification and quality attributes. Process validation ensure that the quality is built into the product during the manufacturing process rather than being tested into the final product. Types of process validation. There are four main types of process validation. First, prospective validation. Second, concurrent validation. Third, retrospective validation. Fourth, revalidation. First, prospective validation. Definition. This is the validation conducted before the new product is released into the market. It is done during the development stage or before commercial batch production begins. Purpose: to ensure that the process will perform as intended and produce consistent and reliable results. Activities involved: defining critical process parameters, critical quality attributes, performing risk analysis, manufacturing and evaluating a number of pilot scales batches under the defined condition, collecting and analyzing data to confirm that the process is capable. Example: validation of the manufacturing process of a new tablet formulation before launching it in the market. Concurrent validation. Definition: validation carried out during actual production of commercial batches intended for the sales. Purpose: used when it is not possible to perform prospective validation due to the urgency of the product supply or when a product is already being manufactured but not yet validated. Activities involved: monitoring and documenting all the aspects of the production process in real time. Data from in process, control, and finished product testing is analyzed. Used particularly for the product where there is limited markets or in emergency situation. Example: a life saving drug needed urgently is validated concurrently while producing commercial batches. Retrospective validation. Definition: validation of manufacturing process based on historical data of already marketed product. Process: to validate processes that are already in place and have a history of satisfactory production. Activities involved: reviewing the production record, batch manufacturing record, quality control result, analyzing data from 10 to 30 past batches, identifying trends, and confirming consistency and reliability of the process. Limitation: cannot be used for new products, less preferred today due to the focus on the real time monitoring and control. Example: a drug that has been manufactured for a year without any major changes and has shown consistency quality may be retrospectively validated. Fourth, revalidation. Definition: repetition of the validation process to ensure that the changes in the process, equipment, or a product do not negatively impact quality. Purpose: to confirm that the validated state of the process is maintained over time or after changes. Types of revalidation. First is periodic revalidation, done at regular interval. Second is a change related revalidation, which is done after any major changes in. So, following are raw material, manufacturing equipment, process parameter, batch size, facility, or the location. Example: a change in the granulator or a dryer used for the tablet manufacturing would require revalidation of the process. So, let's come to the conclusion. Process validation is essential in ensuring that the pharmaceutical product meets required quality standards and are safe and effective. The type of validation chosen depends on the state of the product, availability of the data, and regulatory requirements. Proper process validation not only ensure the product quality but also improves the manufacturing efficiency and minimize risk associated with the production. What is SOP and quality audits? Standard operating procedure means SOP. Definition of SOP, a written set of instruction describing step by step how routine activities are to be performed. Importance: ensure consistency, quality, and compliance in the pharmaceutical opera. Key components of SOP: clear language, sufficient details, abbreviation, and acronyms. SOP preparation process prepared by respective departments with expertise. Review process: QA department checks for the CGMP compliance. Approval: signed, dated, and authorized by the senior personnel. Common SOPs in manufacturing: personnel SOPs, facilities SOPs, material SOPs, productions SOPs. Quality SOP quality audits. Purpose: to verify adherence to the quality system and ensure the CGMP compliances. Type of audits: internal audits, external audits, regulatory audits. Quality documentation pyramid: quality policy, which is a manual, quality procedure, work instruction, and the records. Third, enlist the steps for the ISO registration. First, set the registration objectives. Second, select the appropriate standard. Third, develop and implement the quality system. Fourth, select a third party registration and Apple. Fifth, perform the self analysis audit. Sixth, submit the quality manuals for the approval. 7. Pre assessment by the registrar. 8. Take the corrective action. 9. Final assessment by the registrar. And 10 is the registration. Fourth, explain hydrolytic resistance test for glass container. Hydrolytic resistance of a glass container. Rinse each container at least 3 times with the distilled water and fill with the same of the fillings volume. Heat to 100 degrees Celsius for 10 minutes and allow the steam to issue for the vent cork. Raise the temperature from 100 degrees Celsius to 121 degrees Celsius over 20 minutes. Maintain the temperature at 121 degrees Celsius to 122 degrees Celsius for the 60 minutes. Lower the temperature from 121 degrees Celsius to 100 degrees Celsius over 40 minutes, venting to prevent the vacuum. Remove the container from the autoclave, cool, and combine the liquid being examined. The measure the volume of the test solution into the conical flask and titrate with the 0.01 molar HCl using the methyl red as an indicator. Perform a blank with the water and the difference between the titration represent the volume of HCl consumed by the test solution. So here the table is referred by that. Define recall and classify it. Drug recall. Drug recall refer to the action of removing or withdrawing a batch of a product from the distribution or the use to be returned to the manufacturer. Second, the action is generally done in case where the deficiency are discovered in the safety, quality, or the efficiency of the drug. Third, it is important to note the product recall does not include the normal removal of the product that has passed their expiry period. Fourth, the Organization of Pharmaceutical Producers of India defines recalls as an action taken to resolve a problem with the therapeutic goods for which there are established deficiency in the quality, efficiency, or the safety. Types of recall product. Recall may be of two types. First is a voluntary. This refers to the situations where the manufacturer decides on their own initiative to recall the products where the safety, efficiency, and the quality of the batch is in question. Batch does not complies with the regulatory requirements during stability study done in the post-marketing phases, as in-house investigation reveals a failure that may have an adverse effect on the quality of the batch of a product that has already been released for the distribution. Market complete. Investigations show that the entire batch of the distributed product is defective. Visual inspection of retained samples show the evidence of the deterioration that has the impact on the product quality. Pharmacovigilance reports indicate a serious safety risk to those taking the medicines. Second, situatory recall. These are the recall mandated by the drug regulatory bodies at a central or the state level for the one of the following reason. First, the product violate the law. Example, it is not of standard quality. Second, the formulation contained the banned drug. Third, the labeling of the product or the promotional material violate the law. Fourth, the product is found to be contravene provision of the schedule. The reasons for the product recall. First, the potentially dangerous, serious product quality issues that have comes to the light through the complaint or the other means. Second, mandatory regulation have been violated and comes to the notice of the regulatory agency who then order a recall. New information that comes to light after distribution of the product indicates it is unsafe or ineffective or dangerous. Most common reason for the drug recall: CGMP violence, microbial contamination in the non sterile product, failing dissolution test requirement, degradation product or impurities, lack of efficiency, labeling error, lack of assurance of the sterility, misbranded drugs. Recall classification. According to FDA classified the product recall depending on the health hazard caused by the product. Class 1. It is a situation in which there is a reasonable probability that use of the exposure's products will cause serious health consequences or the death. Example, pathogen in ready to eat the salmonella high level of heavy metal. Class 2 is a situation in which the use of exposure to the defective product is not likely to cause any adverse health consequences. Here, the recall is executed until the wholesaler level and the time limits of up to 30 days is permitted.